Hello, everyone. Thank you for joining us today. Very happy to see a full group of people. Welcome again to our Fall 2020 Clarkson Ignite Speaker Series. I hope all of you enjoyed your week off if you took it, and thank you for joining us again. If this is your first time this semester, we want to welcome you as well. We've had a great 6 talks so far on 2 of the UN Sustainable Development Goals, Goal 11 and Goal 3. And now we're entering our final mini-series on goal number 6, which is water and sanitation. And for this mini-series, we're featuring a specific project on campus focused on harmful algal blooms. So to start off this series, we're going to welcome Dr. Michael Twiss, who will be presenting remotely on harmful algal blooms. Dr. Twiss's research focuses on aquatic biogeochemistry, and he has published over 75 peer-reviewed scientific manuscripts. Over the past 7 years, he has increasingly become involved with the Great Lakes environmental policy. He is an appointed member of the Great Lakes Science Advisory Board of the International Joint Commission between Canada and the USA, a past member of the Great Lakes Advisory Board on Science and Information Subcommittee of the US EPA, and is currently a special government employee of the US EPA. As an active member of the IJC Science Advisory Board, Twiss is presently co-chair of the Great Lakes Early Warning System Phase 2 project and is a core member of the Great Lakes Science Plan Working Group. Twiss was recently elected to serve as the US co-chair of the Smart Great Lakes Initiative, Great Lakes Observing System, and is on the Fulbright Specialist Program roster. For today, we're going to talk more about his current involvement in collaboration with Dr. Yang Yang and Dr. Stefan Grimberg on a Center for Excellence Healthy Water Solutions project involving cyanobacterial blooms. And Dr. Yang Yang will be speaking next week on this topic and more. So if you find today's talk interesting, I highly encourage you to join us next week. So with that, I will pass it on to Dr. Twiss. Dr. Twiss, thank you for joining us today.
Thank you, Matthew. Welcome, everyone. Good afternoon now, and I hope you had a good day voting, preparing to do so. What I'm going to do, as Matthew mentioned, is speak to you about this topic, which is the whole concept of harmful algal blooms. And so you're probably wondering what they are. And so my purpose today is to actually give some background into what we call harmful algal blooms. So what are the causes of them? What are the organisms that are involved? What do we know about them? And so I'm thinking about the Ignite program here on campus. And the whole idea is you have to sort of understand something before you build something. And so I'm going to just step back a bit, though, and make you think about these types of organisms and the situations we currently find ourselves in with respect to these organisms, and then hopefully help you guide your decision. And so when you have a decision to make, it's something that you need to be informed about. And so that is how I'm interpreting this, this Ignite. All right, so here's a little story, and if I'm not eloquent enough, you can just read the sides. Essentially what it is several years ago, there was a crisis in Toledo, Ohio, where over a 3-day period, they shut down the water supply to a city of 400,000 people. And this was traumatic to the people there. You can imagine if we no longer had water to drink, it would be not very long before all your water was sold out of the stores. And that's essentially what happened. And this is in the summertime. And so the cause of this was that Toledo, Ohio, this large city, gets its drinking water from Lake Erie. And there was a cyanobacterial bloom occurring in Lake Erie. And the concentrations of the toxin that's produced by this organism reached elevated levels, which is harmful. And so they had to shut down the water supply to prevent human tragedy. And so this cost $65 million over a 3-day event. And so that is very expensive. And people were shocked. The governor was just about ready to call FEMA to come in and help. And a lot of people were quite surprised by it. with the exception of my colleagues at Bowling Green State University and the University of Toledo Great Lakes Center, because the year before, there had been a similar occurrence in a place called Carroll Township. Now, Carroll Township is pretty small. And so not a lot of people, not a lot of voices. And they were actually able to shift their water supply. So it didn't really cause much of an impact. But the warnings were out there, but no one listened. And so that is actually one of the problems here, is when we don't have enough people listening to those that are marginalized in our society, then what can happen is we can get worse impact from these kinds of occurrences than would be expected just based on the straight impact alone. So in other words, it compounds itself. And so there's the whole idea of social environmental injustices, which is a United Nations Sustainable Development Goal. I heard Matthew mentioning them. Moreover, something that I'll mention later on is that these harmful algal blooms are increasing across the Great Lakes region. And I'm going to bring out the Great Lakes examples several times in this talk. That's because that's where my expertise lies. And it provides a good example of the kind of difficulty we find ourselves in with respect to cyanobacterial blooms. And one thing that's of interest is that algae concerns are most cited by Great Lakes Indigenous respondents. So the International Joint Commission, Canada, United States, did a survey back in 2018, and the number one concern for them was harmful algae, which I found surprising. I'm not expecting that. I thought it would have been contaminants in fish. So here's some perspective. Okay, these are microscopic organisms, yet we can see them from outer space. Down on the right-hand side here, down below, this is Lake Erie. This is 200 miles long. It's a great lake. Okay, it's the one that's the furthest south. Toledo is right over here where I've got my the pointer. And what you see here is a large cyanobacterial bloom. And it's going out into the central basin, being carried by the wind and the currents. And this is cleaner water coming in past Detroit from Lake Huron. This is a boat. You can see this little boat right here. It looks a little skiff. There's two people in it, and they're cutting across this bloom. And you can see some of it's older, so it started to photo degrade in the sun. And you also see the boat wake is actually parting it. And so it's mostly at the surface. And that's an important thing because most of the time we see these blooms because we see the surface of the lake. It's hard to see into lakes. Here's a close-up showing what they look like. Sometimes it looks like there's paint been spilled on the surface of the water. That's because these cells are actually buoyant. So on a hot day in the west basin of Lake Erie, you're out there with your family, you're thinking about going to the beach, and this is what you see. Not unexpectedly, there's not a lot of people at the beach. Some people are actually hazarding to go in the water. I wouldn't put my child in there because if they went underwater, I would never be able to find them. This is a degraded situation. And so the important thing about water, and we heard a hockey speaker last week talk about the importance of water globally and personally. And if you think about the Great Lakes, we use it for industry. We use it for transportation. We use it as a source of drinking water. We use it as a place to dump our waste in the form of treated sewage. We use it for recreation. We use it for sort of well-being and for spiritual reasons. And so it's a really important resource. And so for us to be faced with something like this can be quite devastating. So here are the Great Lakes. I'm sure you're all quite familiar with them. Here we are over in Potsdam, center of the universe, right about just south of Messina. Okay. And what I've circled here are all the places where cyanobacterial HABs are occurring currently across the Great Lakes. Now, why are the Great Lakes important? Well, there's 35 million people that live within the basin, which is the green section around the lakes there. So the watershed. If we include the province of Ontario, Quebec, and the 8 US states, combined, the third largest economic bloc in the world. And they contain 20% of the world's surface fresh water. And so, as I mentioned before, we use that as a drinking source, we use it as a waste disposal. So it's a really important resource in which to manage and to protect. And so the places where these are occurring this is the Bay of Quinte region over here near Kingston. It's a wine growing district. Down here near Rochester, Hamilton Harbor, Lake Ontario, Honey Harbor up here in Georgian Bay. This is again like affected by cities. This is the West Basin. There's Toledo, Saginaw Bay near Chicago and the Chicago River, Illinois Waterway, Green Bay, Wisconsin. These are places where we've had reoccurring blooms. Also over here in Duluth, near in the harbor, the mouth of the St. Louis River, and over here in this portion of the Madeline Islands region of Lake Superior. So you don't usually think of Lake Superior as a place where there's blooms. But what's happening though is that these blooms are becoming more widespread as time goes on. So here's this region near the Madeline Islands rather, in Lake Superior, and it's beautiful. If you've ever been to Lake Superior, it's astounding. The water is so clear, you can see easily 30 feet down, and it's clear water, and you can stick your head in the lake and you can drink it for the most times. However, there has been just recently some cyanobacterial blooms occurring in Lake Superior. And they normally have been occurring in just like embayments where there's cities and there's high agricultural activities. But what's been happening is increasingly there have been incidents of it occurring in regions where we're not really expecting them to occur. And that's a real big problem for Great Lakes limnologists like myself, because we're wondering, how can you get these cyanobacterial blooms occurring in places which are naturally quite nutrient poor? And some of the reasons are due to invasive species. Zebra mussels have been re-engineering the phosphorus cycle in the lakes, and it's tending to keep the nutrients on the near shore. And where do people interact with the lakes? It's not out in the middle of the lake, it's near the shore. And so that, along with climate change, are the most important factors which are affecting the prevalence and the occurrence of these blooms. And so here's the bloom here in 2018. You can see that water clarity has gone down to less than a foot, from 30 feet to less than a foot. So quite an issue here. It's also global. We've been quite preoccupied with COVID in the past year. And so you may or may not have encountered this news item. But they're discovering in the Okavango Delta in Botswana a number of dead elephants, and they're saying, oh, this must be poaching, but it wasn't poaching. They found them dead near water pools, and those pools were full of toxic cyanobacteria. So they're drinking the water from these pools, and they're dying. And they contain a third of Africa's elephant population, and they lost 330 just during the month of May and June of this year. And the cause was toxicity from cyanobacteria. So that's now. So we know that they're here now. But they've been here in the past as well. In fact, records of it. This is a quote from the Bible. And so people recorded these events back in the day, and they've carried that on through the Christian Bible. It's also mentioned in the Quran, and it's also mentioned in the Torah. And so this was God speaking to Noah, saying that I'm going to get at the Egyptians, essentially. I'm going to whack them. He's going to strike the water of the Nile, and he's going to change it into blood. And the fish will die, and the river will stink, and the Egyptians will not be able to drink their water, and they will release the children of Israel from bondage in Egypt. So you may be aware of this story. So it's one of the up to 10 plagues, depending on which book you're reading, which occurred in ancient Egypt. And so the record shows from these historians that wrote this down in this holy book that the water did turn red. It turned to blood, essentially. Now, there's some evidence that this actually has some ecological and geochemical sort of explanation. And so there is evidence that there was a climate change occurring around this time in Egypt. So Egypt was becoming more dry. So it was experiencing a long-term drought. And they know that from the geochemical evidence found in the form of oxygen isotopes, which are found in stalactites and stalagmites inside of caves in the region. And so those reflect the amount of the proportion of precipitation that's occurring from evaporation from the oceans. And they know that the climate change was occurring and was getting drier. And so this would have slowed the water flow in the Nile River. And as ecologists and microbial ecologists, we know that that has an impact on the plankton community. Several authors have concluded that there have been microbial changes in the river would have occurred. Planktothrus rubescens, which is a cyanobacteria, it produces the hepatotoxin microcystin. We know that's deadly. Hematococcus pluvialis is another cyanobacterium. And then we have Euglena sanguinea, or blood red, essentially, produces this very potent fish toxin. And so there your fish dying, there's your color. And so there's an explanation for this. As I mentioned before, the spread of cyanobacterial harmful algal blooms has occurred increasingly over the past few decades across the Great Lakes. And this is almost prophetic to me because our indigenous communities across the Great Lakes are actually, many of them are Christian and they'd be quite aware of the Bible. And so they are cognizant of the fact that there have been these kinds of things that have happened in the past. And I was just made the connection when I was preparing this talk that is perhaps why they were so concerned about harmful algae rather than things like toxic contaminants, which are much easier to sort of identify, do fish advisories. If something is affecting the water itself, then we've got big problems. And so this is, to me, is like an early warning of our situation that we find ourselves in today. So what are these things that cause these plagues? So I'm going to use the term algae, and it's quite a, it's like saying almost like, car. There's different types of cars, and there's different types of algae. And so I'm going to talk a bit about these things called the cyanobacteria. Then we'll discuss a bit about the causes which make them proliferate, and then finally how we can protect ourselves. So what are these things? Well, the next few slides I've grabbed from the Association for the Sciences of Lunology and Oceanography. I've been a member since '92. And Frances Pick here is at the University of Ottawa. She's spoken at Clarkson before. She was my TA when I was an undergrad, and she's now a professor at the University of Ottawa. And we were academic siblings, actually, because we shared the same professor, Dr. Cheshire Alvaiko at the University of Toronto, where she did her PhD. I did my master's at the time. And so she's an expert in this area. So I'm thanking her for her sharing of these slides with me, just a few of them. Okay, so what are these cyanobacteria? They are prokaryotes versus eukaryotes. Okay, so they are prokaryotes. So they're a type of bacteria. That's hence the name cyanobacteria. The cyano comes from the fact that some of them give a blue-green color. So there's your cyano color. And they're different than eukaryotes. So this is a eukaryote cell. So the cells in our body are eukaryotic because we have a nuclei. Now, some plant cells and other algae are eukaryotes because they contain within them a chloroplast, which evolutionarily originated as a cyanobacterium. So kind of interesting. So cyanobacteria have actually existed for greater than 50% of the lifetime of the Earth. So there's about 4.8 billion years old, and cyanobacteria have been around from the Precambrian era, so about 2.7 billion years ago. And what makes them important is that these are the first organisms that photosynthesized, and in doing so, released oxygen. There's another type of bacterial photosynthesis that does not release oxygen, but these ones did. And so they have been responsible for changing the geochemical conditions on the surface of the Earth, and the composition of the atmosphere around us is much in part due to the evolution of these prokaryotes called cyanobacteria. So as I mentioned before, they're microscopic. They're unicellular. They grow in colonies. They can be filamentous and have all sorts of different types of cells associated with them that do various sort of special functions. And that actually is important because the small ones can be eaten by others, but when they start to clump up and become colonial or filamentous, that actually prevents them from being eaten by these little shrimp-like organisms, which we call zooplankton. So that's one of the factors involved with them being able to proliferate. They produce toxins, a whole slew of them. And they have a wide range of structures. And the big ones are your microcystins, your anatoxins, saxitoxins, and cindolospermopsins. And these are the kind of toxicities that they cause. So a hepatotoxin essentially turns your liver into chocolate pudding. Okay, it starts to degrade your liver, and so you will die. Neurotoxins obviously affect nerves, and then cytotoxics are ones that break apart cell membranes. Cell membrane dies or breaks, the whole cell dies. And they are toxic at relatively low concentrations. Now, what's the purpose of these toxins? And so there are a number of hypotheses, and there's various evidences that show that they are a defense against grazing. So they can inhibit other organisms, so they won't be eating them. Allelochemicals, they can be used to compete against others, much in the same way that antibiotics are used by fungi and bacteria to outcompete other fungi and bacteria in their environments. They are able to establish what's called a quorum, so they know how many other organisms are present in their environment. Some of them are used for metal acquisition, so they can grab metals and hold on to metals. And they're also, in some cases, used as reserves of nitrogen. And so, for example, microcystin is, I believe it's about 20% by weight nitrogen, and that's a nutrient. And so it might be a way of storing the nitrogen in their cells. And some way of countering environmental stresses. So they respond to stresses. These toxins, for example, in the case of microcystins, 60 people died in Brazil back in '96, where they were taking their water source and they were not cleaning it, like UV oxidizing it. And there was a cyanobacterial bloom in their reservoir. It got into the dialysis water that they're using for transferring salts out of people. And as a result, a number of people perished. There have been examples in New York State of people playing in lakes with their dogs, and their dogs have died after licking this material off of their fur. So where do we find them? We find them pretty much in anywhere there's water. They're most diverse in fresh waters rather than the oceans, but you can find them in soils as well. And some of them are actually pink colored. When I was doing my PhD, I got a cyanobacterium from Gary Faunsteele at the NOAA lab in Ann Arbor. And he sent them to me, and they were pink. And I thought, oh my gosh, these are contaminated. But they weren't. They just tend to have more of a phycoerythrin pigment than a phycocyanin pigment, which is the blue-green color. So the other thing here is that they create what are called blooms. And so when you think of a bloom, you think of, wow, this just appears. They grow so fast. But that's a fallacy. They don't grow fast. Cyanobacteria are notoriously slow-growing organisms. And so you might think, how do they appear so quickly in the environment? And why are we so concerned if they're such slow growers? But what happens is that they become visible, and they can become visible rapidly. It doesn't mean they're growing quickly. Blooms can persist for days up to months, or in some cases, in tropical regions, they can be constantly present in a lake, so very high concentrations of cyanobacteria. And so concentrations that are greater than 50 micrograms per liter of chlorophyll A are considered to be scums and essentially a bloom. What you're seeing up here in this picture, this photograph I've got in the backdrop, that's hundreds of micrograms of chlorophyll A per liter. So this graphic up on the left shows us the some situations. Over here, we've got stability of the water column. So if you're in the North Country just recently, it's been cold, it's been windy. What's happening this time of year is that the lakes are being destratified. They're cooling off, they're becoming isothermal, the same temperature from top to bottom. And then when the wind blows, the lakes turn over. So that's a situation of full mixing. So in that situation, you're going to have cyanobacteria dispersed throughout the water column. But when it's stratified, for example, in the wintertime, it did stratify in the wintertime, but in the summertime, mostly when cyanobacteria will predominate, what occurs if there's not a lot of wind energy, and what will happen is under high light levels, is that the cyanobacteria will float to the top, and I'll talk about that in a moment. So they actually accumulate at the surface. At lower concentrations of light, they can actually be down lower in the water column. So we can find them throughout different parts of a lake, depending on the conditions that are prevailing. And so what are these traits that cause them to dominate and create what we call blooms? Well, first of all, some of them create these very large colonies. So microcystis is less than 2 microns in size, but they clump together to make clumps which are visible to your eye. And some of them are actually, some other species are filamentous, and so they can't be eaten by other grazing organisms. So they're not very edible in that sense, physically edible. They can tolerate extremes in solar radiation. So because of the pigments that they have in them, they can protect themselves against high light. And at low light conditions, they have extra pigments that help collect extra photons, and they're not so abundant. So they've got this flexibility inherent in them. They have the ability to store nutrients, as I'll mention in a moment. They're also capable of doing what's called nitrogen fixation. And this is remarkable because most of the atmosphere around us is dinitrogen gas, N2, and that is inert, except under very high temperatures, except that some bacteria and some cyanobacteria are able to take nitrogen and convert it into ammonia inside of their cells. And to do so, they need a lot of trace metals like iron. They're also able to regulate their own buoyancy. And so how were these bacteria in the backdrop here able to end up on the surface of the lake? That's because they have what are called gas vacuoles inside of their cells, which allow them to become positively buoyant. And they also have these benthic resting stages. So around this time of year, many of the cyanobacteria will start to senesce. They sink to the sediments. In the springtime, after the ice is gone, the winds cause the mixing to occur and they re-inoculate the entire water column. And so they are really good at taking over environments that are favorable to them. So the main sort of nutrients that photoautotrophs need are carbon dioxide, which they find in the water, but they also require phosphorus and nitrogen. I just mentioned nitrogen before, a number of them have these specialized cells called heterocysts, which are able to fix nitrogen to convert nitrogen gas into ammonia. And they're also able to store nitrogen in the form of cyanophysin. So they can store their own nitrogen inside their cells. And that is unlike other eukaryotic cells. So they have an advantage there. They're also able to accumulate phosphorus and store it as a polymer called polyphosphate. So it's not an energy storage, but rather it is a nutrient storage capacity. So these cells, in the availability, if nutrients are highly available, they have the ability to hyper accumulate these nutrients. Moreover, many of them produce what are called siderophores. These are organic compounds which are secreted and remain on the outside associated with the cell, and they have a very high complexation capacity for iron. And so if you have siderophore in the presence of rust, it'll dissolve the rust. It'll just grab it. They've got a log stability constant of greater than 10 to the 20. And so they need that for nitrogen fixation. On the right-hand side here, we see a electron photomicrograph of a cross-section of cells. And you can see the vesicles here, they're shaped like spindles. So look like long sort of spindles. And they all go together. And then in this case here, you see them sort of cut. So you can just see the tips of them. And they actually cause them to be positively buoyant. Now what happens is a cell at the very surface of a lake will begin to accumulate carbohydrates and polyphosphate granules and actually ballast them, and they sink down below. Then they use up those nutrients through respiration, and they divide. And so what happens is they become more positively buoyant, and they start to rise back up to the surface. And so that's their cycle. So they have that buoyancy that's due to the gas vesicles, but it's regulated by the availability of light and the availability of nutrients. So briefly, in summary, these blooms of these types of algae are actually technically these prokaryotic cyanobacteria, so pretty primitive life forms that have been around for a very long time. They're oxygenic photoautotrophs, meaning that they produce oxygen. They do serve a role in environments, and that they are food for some types of organisms. And in fact, if you're in Lake Erie like this, it's actually really good fishing. There's lots of walleye down there. They like it when it's kind of dark with these blooms covering the top. And some of this energy gets transferred down into the food chain. Not all of it, though. What causes these blooms to occur? A lot of factors, as I mentioned before. They're buoyant, so they get to the surface. And when they're at the surface, that's when we see them. And that's when people complain. And one of the most difficult things that the New York State Department of Environmental Conservation is faced with is that they have this hotline. So at first, when there were blooms, they would all get in their trucks and they'd rush off to the lake, and then they get there and it's gone. That's because they'd rise up, the wind would blow, the waves would start, they get dispersed and trained back down into the lake. Or they get blown to the other side of the lake, where this person who called no longer can see them. And so that is a big challenge for people to understand that these blooms will come and go. Most of the time, the algae are actually down in the water itself and only appear when they rise to the surface. And for the most part, they're difficult to eat, and they have this ability to take on many nutrients through siderophores, accumulation of phycocyanin, the accumulation of polyphosphate, the production of siderophores. So the next question is, how do we protect ourselves from them? So at this point, I'll just pause for a moment and ask if there are any questions, Nathaniel. Are there any questions that people want answered from the point of view of clarity at this point?
We do have a few questions. So we'll start first. Jake asked, and you sort of alluded to it, but if you wanted to elaborate, that would be great. He asked, like the elephants in your example, can waterfowl and fish also die from cyanobacteria ingestion? Has this been documented? If so, how common is it? So have there been any large-scale die-offs in the Great Lakes region for any popular wildlife?
Yeah, with respect to cyanotoxins, no. Clostridium botulinum, yes. That's caused die-offs of both fish and of fish-eating birds. We do know that microcystin does get into fish flesh, but not at high concentrations. And it's actually something that the US EPA currently does not monitor, although that might change with the next round of funding. Good question.
And then, sorry.
Go ahead.
And then Maria asked, what creates the increase in nutrient availability that allows these blooms to occur? And does there have to already be cyanobacteria present in an ecosystem for blooms to occur, or can they come out of nowhere?
Well, they have to be present somewhere. So they are often in resting stages, And when the nutrients are available, then they can begin to grow. One of the issues that we currently face with, which is probably the most readily manageable, is nutrient management. And so this is things like wastewater treatment, making sure we have buffer zones around lakes and rivers, making sure we have up-to-date septic systems, making sure we don't over-fertilize. So if we can control the nutrients getting in, that's a big first step to controlling cyanobacterial blooms.
Now,
Thanks,
if these strategies are operationalized properly, is it enough to control the blooms? Like, is it just a funding problem, or are there better technologies or strategies needed in addition to the ones that are available now?
There are some big problems with our abilities to control cyanobacterial blooms. One is that they're really well at growing even in lakes that have relatively low nutrients. Controlling nutrient is identified as being the top, the first sort of line of defense for controlling the blooms themselves. In fact, Lake Erie, the Canada, United States have decided by 2025 to reduce phosphorus inputs into Lake Erie by 40%. 40. And that's in 5 years. That's going to be costly. But look at what you're seeing there. That's the West Basin of Lake Erie. Next question. Are we good?
We're good so far if you want to keep going.
All right, thank you. So the question is, how do we protect ourselves? Controlling nutrients is one way. Excuse me. The big problem, though, is global warming. And there is evidence across the globe that lakes are warming in response to increasing atmospheric temperatures. Some are actually cooling. So on this figure here, which is from O'Reilly et al., 2015, you'll see a couple places where there are some lakes which are actually cooling, depending where they are because of the change in temperatures. It's relative increase or decrease. If you notice the magnitude here, though, this is half of what the scale is over here. So on a global sense, most lakes are warming. And what this causes is a reduced period of ice cover. And so lakes are taking longer to freeze over. Around here, Lake Azonia, for example, starting to freeze over, just after Christmas. Before it used to freeze over much earlier. I speak to a gentleman who's helped our limnology class up there repeatedly, and he's kept records. And in fact, we, in our limnology class, have sampled there in the past. And there's a thesis in the Clarkson Library from 1975 where someone was looking at phosphorus in Lake Azonia. And the lake temperature on average has increased 2 degrees Celsius since 1975. That's just one example. Your example is right across the globe in front of us. And so the problem with that increase in ice-free condition is that it increases the opportunities for cyanobacteria to thrive. They do tend to grow at warmer temperatures. And so even though they grow slowly, they prefer to live at warmer temperatures than colder temperatures. And so this is a big issue for us. I have been measuring water quality in the St. Lawrence River using remote sensing techniques since 2014. We have stations here at the 32nd turbine, 17th, these are both in New York, and over here's Canada. My colleagues at the River Institute have operated this unit number 1 here a few years afterwards. Over here we see temperature and cyanobacteria. The cyanobacteria are the blue-green cyanal peaks here that we see. And here's the limnology class out in 2014, collecting plankton tow from about 20 meters, 60 feet in that little net. And that's all the cyanobacteria. So there's actually a bloom occurring. But if you look in that river at that point, you can see down 6 meters. you can see almost 20 feet down into the lake, into the river. So it's relatively clear, except those cyanobacteria are there. So naturally, cyanobacteria tend to accumulate and then manifest towards the end of the growing season. And this is called the tortoise hypothesis, where you get a tortoise in your hair. Some algae get out they just go for it in springtime and they're done, like diatoms. But cyanobacteria tend to, because I said before, they're slower growing, takes them more time to manifest. And there's other ecological reasons that I'm not going to get into. A couple things that are interesting here in 2017, this was that big flood year. And so the bloom is not as apparent. It's suppressed because it was diluted out. The following year, in 2018, was the warmest on record. And so this is the highest temperature ever measured. It broke 24 degrees Celsius. And here's the blooms of cyanobacteria that are increasing in the lake, in the river rather, at that time. And speaking with colleagues, Professor Holson here at Clarkson University in civil environmental engineering, he's a scuba diver. And he was mentioning that the water was much more turbid that year. Cyanobacteria, warmer temperatures. So this is common now, unfortunately. So how can we protect ourselves against them? I mentioned earlier that reducing eutrophication is probably the #1 approach. And this is one that the International Joint Commission has led the Canadian and US governments to agree that that is what is most doable. The other thing is you can develop early warning systems to avoid exposure. This is something I'm getting increasingly involved with. And so when I hear like people worried about this that are close to the ground, they're worried about cyanobacteria, then I'm listening. Toledo now has early warning systems. They have sensors on their water intakes. And so they measure meteorological conditions and water quality, and they know when to shut off the water so it doesn't get into their system to contaminate it. And so that's their early warning system that they're using. And so there's also what are called algae watches, cyanowatches. These are operational models that have been established, and you can go online, you can get them mailed to you every week. They'll tell you what the bloom is, what the bloom's expected. In the springtime every year, NOAA puts out a prediction for a bloom for that coming year. It's not bad. It's not perfect, but it's a beginning. There are also some engineering fixes, and here at Clarkson, of course, we like hearing about those. And so we can actually destroy cyanobacteria, we can disturb them, and there's a number of different ways to do that. One of them is to use alum and iron oxide hydroxide to actually sorb onto the phosphate and trap it in the sediments. And by doing so, you prevent them from inflating in the water column. Well, that's good, provided you're also reducing eutrophication. So it's not going to be sustainable if you do that and you still don't work on your septic systems in your region, okay? Because you're still going to have to be doing that. So in other words, unsustainable. But sometimes it's good to sort of pass that tipping point where if a lake goes hypoxic, then you get internal loading of phosphorus inside of a lake, and that'll keep on triggering cyanobacteria. So sort of capping it is actually one feasible way. And all of these approaches here are only sustainable, I would argue, for lakes that are used as a drinking water source. If it's a lake that it's just because a bunch of people live on it and they say, oh, we just like it looking differently, you got to question whether or not it's worthwhile. Then we have things like destabilizing the water column, so taking away stratification, so mixing, so you prevent the bloom from occurring at the top. And to me, that's a nice big Band-Aid, because you're still going to have cyanobacteria in the water column. They're just going to be distributed. There are also different ways to keep them away from various places. So if you've got a water intake, you can actually make bubble screens or physical screens to keep the water from moving near your water intake. And again, these are very expensive engineering fixes. And again, most useful if they are for protecting source water for drinking. And finally, there's another bunch of chemical approaches. So hydrogen peroxide is very effective against cyanobacteria, more so than eukaryotics. And that's because eukaryotic cells produce their energy inside of organelles inside their cells, like mitochondria or chloroplasts. Whereas the cyanobacterium produces its energy on that outside membrane. And so it's very sensitive to changing redox conditions. Hydrogen peroxide is deadly to cyanobacteria, but less so to eukaryotic organisms. But again, are you going to add peroxide to the west basin of Lake Erie? Ultrasound has been used to break apart cell gas vesicles. And electrochemical oxidation, which you'll hear from next week, is a way of producing oxidative radicals, and that's able to, again, kill cyanobacteria. Would be a very good approach to treating waters that are used for source water for drinking. So the big issue here is how do you decide where and when to spend your resources? So we have a limited amount of resources in our society. We have a lot of money we do, we're a rich country. But we have to be very prudent in how we spend that money. And so it becomes these questions of divisible and indivisible goods. How do you share certain things like fresh water? How do you treat one part of a lake without treating another part of a lake? Which lakes do you treat? Which lakes do you not treat? Who decides that? We're making decisions today, as you know. But I didn't see any concern about cyanobacteria on the ballot. However, we do know that some people are concerned about things like climate change. And climate change has actually been identified as the primary driver that's causing cyanobacteria to proliferate. We're finding cyanobacteria proliferating in inland lakes that are away from nutrient sources. And so the answer is being able to control climate change. And this is a very challenging problem to solve. There's no quick techno fix to it. It's very difficult. And I think that folks at Clarkson are well suited to participate in that challenging question. And so I'll leave you with this. When you choose to make a decision on how you're going to work on this problem, if you choose to, think about the most sustainable way to approach the problem. So at that point, thank you for your attention. And if there are any other questions, I'll welcome any.
Great. Thank you, Dr. Twiss. At this time, we do have about 10 more minutes slated. So if there are any audience questions, please enter them in. In the meantime, I do have a prepared question I'd be interested to ask. You know, you talk a little bit about the mitigation or remediation costs. You talked about the, the fact that some of these remediation plans are basically slapping a Band-Aid on. What are the, are you equipped to talk a little bit about the difference in the cost to mitigate or remediate these algal blooms versus the cost of the economy if we did nothing? For example, you talked at the beginning of your presentation about the cost for 3 days in Toledo. Mm-hmm. How does that kind of extrapolate across the entire region? Do we know?
I'm not prepared to answer that question.
Okay.
I don't have those numbers at hand. So
it did look like we did have one more audience question from Jake again. He asked, many cottages on lakes have water intakes between 7 and 15 feet. Will mycostacins be a threat at that level? And are they capable of being present even if a bloom is not visible at the surface?
Yeah, good question, Jake. So yeah, 7 to 15 feet would be pretty shallow. In lakes around here, depends on the morphology of the lake and the size of it. Chances are that's still in the, what we call the metalimnion. And so that's where the water is cooler, but it's not the surface. And so generally, when cyanobacteria do accumulate, they'll tend to accumulate at the surface. And so if you're drawing water from further down, odds are you'll be able to miss them. But what you can do, which would be advisable, is to just filter them, pass your water through a fiberglass wool insulation or sand filter. Actually, the Egyptians, if you read the Bible, the Egyptians were taking their water from the wells beside the river because they're sand. And that was essentially a sand filter that they were doing it from. Because if you try to do something like add peroxide or bleach, what can happen is you'll break apart the cells and you release microcystin. And so that's, then thermodynamically, it's more challenging to get that out of the water than the particles themselves. So if you can let the particles settle, then you'd be in good stead. So but first off, if you suspect that there's cyanobacterial bloom in that lake where there's a cottage, you should notify the New York City DEC about it. They can come in, take a sample, and tell you if there's toxin in it or not. Rule of thumb, though, just avoid it. Cyanobacteria do produce geosmin and methylisoborneol, two compounds which give a sort of a soil flavor to water. And so humans can detect that at very low concentrations, like 10 nanograms per liter. Women are even more sensitive to it. And they believe that's an evolutionary adaptation. Those compounds aren't toxic, and there's no correlation with toxins, but periodically there could be. And so there's obviously a selection for us being able to detect that. So if the water that you're taking smells like muddy, obviously you're not going to drink it, and that's healthy for you.
Great. Well, I think that those are all of the questions that we have today. So I want to thank you all again for joining us. And we hope to see you next week when Dr. Yang continues this discussion and starts to talk about solutions to both harmful algal blooms and how electrochemical processes can be used for other water contaminants. Thank you very much, everyone. We'll see you next week.